All right, finally, I'm ready. Next chapter, okay, we'll look at fertilization, pregnancy. I'm cutting out the development part because it's getting too late in the semester. I'm, for some reason, behind. I missed a couple of lectures, so, and the development part is a little bit too complex to really do justice anyway. So we'll talk about pregnancy, and then magically there's a birth. We'll talk about the placenta a little bit, but not about all the changes in the embryo that actually builds the body. All right, so what chapter is that? 28. 28. So if you want to have development and all that, you need first to have fertilization, and fertilization begins with sperm penetration. Sperm penetration. That's the first thing that has to happen for fertilization to occur afterwards. The funny thing about the mammalian eggs, when they get ovulated, they're actually surrounded by a lot of stuff. So the egg is not that easily accessible to the sperm directly. So here we have the oocyte. This is actually the secondary oocyte, right? Arrested in metaphase 2. And then it finishes metaphase 2. Here it's kind of starting to, right? They have a little mitotic figure there. When sperm starts to surround the egg and starts to penetrate that oocyte. And the egg is really surrounded by a lot of stuff. As I said, there's this thing here, the zona pellucida, and here it's magnified. This sort of whitish area. This is a reduced egg envelope, like an egg shell. So even though mammals now retain the eggs inside their bodies, and you have internal fertilization, and the embryo and later fetus develops inside mom, this is an evolutionary holdover from the time where everything laid eggs, and we still have this basically reduced eggshell, the zona pellucida. Zone pellucida, translucent, because on the microscope, if you look at an oocyte under the microscope, it looks like a clear ring around it. So the clear zone is basically what that means. But it's a little proteinaceous eggshell surrounding the oocyte. And then in addition to that, you have a lot of the follicular cells, the granulosa cells, come off with the egg as it's ovulated. So all of these are cells from the ovary that stick to the outside of the zona pellucida. And they form what is called the corona radiata, the radiating crown, because again, under the microscope, a lot of times they're sort of layers of cells sort of radiating out from the oocyte. Here they didn't really draw it quite like that, but there's lots of cells stuck to the outside of that egg. So sperm have to be attracted to the egg. There's chemotaxis. And it's still not really that well understood in mammals because of the internal fertilization. It's difficult to study what goes on inside the fallopian tube where fertilization occurs, to actually measure what kind of proteins might be released there from the egg and so on. But there seems to be some chemotaxis, so that the egg releases chemicals that then attract the sperm to it, so the sperm can find it. And they definitely seem to really hone in on it. Then, when the sperm make contact with that, they start to make a hole through the corona radiata, through those cells, to actually get to the egg membrane itself, which is all the way down here. So we have a lot of stuff to get through. So here we're looking at a picture in figure 2 of the approach. Aided by enzymes on its surface, the sperm cell weaves its way past granulosa cells of the granulosa of the corona radiata and makes itself burrows in there. Now, the oocyte after ovulation is viable at the most about 2 days, at the most about 1 day, 24 hours. So between 12 and 24 hours viability for the egg, and sperm between 24 and up to 72 hours viability. So you can have sexual intercourse, sperm is around in that area So it's a little bit bigger window for sperm than for eggs. So if you ovulate anywhere in that 72-hour window, let's say, there's a chance for fertilization. And the sperm has to make it all through all these layers. Here now, it made finally contact with the eggshell itself, with the zona pellucida, and there are very particular sperm cell zona cell interactions. It's almost like a lock and key mechanism, so that only human sperm can actually physically make that connection complete and actually bind to the egg cell. And when it does that, and that prevents potentially cross-fertilization. And again, that's a holdover from animals that used to fertilize externally. So if you're fertilizing internally, that's much less important because you're not going to mate with some other species, right? If you're internally fertilizing, you're mating with your conspecifics, and that pretty much because of behaviors. And think about it more. If humans were just humans only, but think about other animals where there are more species that are closely related that might interact in the environment. Well, why don't they mate, like different kinds of closely related lizards or whatnot? Well, there are behaviors that make sure that only the ones from the same species, and then animals are, courtship dances, that type of stuff. So in internal fertilization, there's a little bit less of this lock and key stuff, but it's still there. So the sperm needs to have receptors that it binds to that actually match that same species, and this happens even in humans. And then what that triggers, when it binds properly to a human zona pellucida, then we have an acrosomal reaction. Okay, acrosomal reaction. This is important. The term acrosomal reaction, and if you remember, the acrosome was that little vesicle that sits like a hat over top of the sperm. So if you don't remember that, go back looking at spermiogenesis, where the sperm turns from the little round cell to growing the tail and all that. So that's where it mentions the acrosome the first time. It's a little cap, a little vesicle that sits over the sperm cell nucleus like a hat, and it contains enzymes that, for the most part, will digest a whole for the sperm through the egg cell membrane, through the eggshell. I'm sorry, not the cell membrane. So the acrosomal reaction. Don't worry about exactly how this happens. So just write down, okay, sperm binds to the zona pellucida, acrosomal reaction happens. What that means is that this stuff comes out of the acrosome vesicle and gets disgorged onto the eggshell membrane. Where does it say that? Acrosomal triggering, acrosomal enzymes from many sperm actually digest wholes through the zona pellucida. So it's not just one sperm having an acrosomal reaction, it's more the ones that come after. Also, they'll dump the contents of these acrosomal vesicles onto the eggshell membrane, onto the zona pellucida, and these enzymes are basically breaking down the zona and digesting a whole through it. So the first sperm that actually makes contact with the eggshell, with the zona, is not going to be the one that gets to fertilize the egg, ultimately, because it takes sort of a group effort. They sort of die in battle. It's the little bit later ones that finally, when the hole is big enough, will actually, oh, okay, now I'm going in. If that's fair or not, you decide. So the first to the egg is not going to be the one that actually gets to fertilize. So the acrosomal enzymes from that vesicle, that little hat over the sperm head, digest a hole in the zona pellucida. And then the sperm, finally one of them is actually going to be able to wiggle through that hole and bind to the egg cell membrane itself. And that's right here. So the oocyte has receptors for the sperm as well. And then when the sperm binds to that itself, it triggers a second reaction. This one is called the cortical reaction. Where's that on here? So fusion, the sperm and oocyte plasma membranes fuse, allowing sperm contents to enter the cell. Block of polyspermy, don't they call it cortical reaction anymore? cortical reaction here. So the final fusion of the sperm to the actual egg inside the shell triggers this cortical reaction, and it's a release of stuff from granules in the cortex of the oocyte. Okay? Cortex is the superficial layer of the oocyte. Cortex is always the superficial area of something. And these cortical granules contain things that make sure only that one sperm that just got in is the one that gets to fertilize that egg. Because you only need one packet of chromosomes, you don't need any more. Okay? Because that would add extra chromosomes and you'd have 3 or 4 sets of chromosomes, and that's no good. You only need the 2. And that is called the block of polyspermy, blocking many sperms from entering the egg. Once that first one gets in, the cortical reaction blocks polyspermy, blocking any more of those sperm to deliver more chromosomes. We only need one set from that, not 2 or 3 or however many. And these cortical granule enzymes and the stuff that's in there, one of the important things that it does, it actually removes all the receptors for sperm. Does it talk about that? The sperm receptors are clipped off. So there are no longer ways for new sperm to actually make contact with the membrane. The sperm receptors are removed. And it releases chemicals that are really attractive to water and suck water into the space between the eggshell and the egg itself. So this will fill with fluid and will remove the eggshell from the membrane of the cell inside of it and make a big gap there. And then it also hardens the zona pellucida so that other sperm around there have not as easy a time to actually digesting holes through it. So there are all these different ways of making sure only one sperm actually gets to fertilize the egg. So there's lots going on with sperm penetration, right? So we have acrosomal reaction. Make sure you understand what that is. That's releasing these enzymes from the sperm to digest a hole through the eggshell. Then you have, finally, one of the later sperms can use that hole then and bind to the egg membrane itself. That triggers the cortical reaction, and that makes sure we do all the different things to make sure that no other sperm are able to do the same thing, blocking polyspermy. Hardening the eggshell, making the gap by filling this with fluid, removing the sperm receptors. Because you only need one set of dad's chromosomes, not more than that. Then we get here, 28.3. So now the sperm has penetrated, and really all the sort of extra stuff from the sperm stays outside. The egg cell in the nucleus with the genetic material goes in. So here we have the sperm nucleus, and the egg at the same time, remember, was stuck in meiosis too, right, in metaphase. Now it's finally going to finish that. So here you see second meiotic division of oocyte is happening. Sometimes the polar body will have a meiotic division, not important. And finally, then you have your chromosome numbers halved in that ovum. And all these polar bodies are just packets of chromosomes that you don't need. And the nuclei swell, so the sperm nucleus will swell up. The female nucleus that results from that last division swells up, and then they're called the pronuclei. Male pronucleus, female pronucleus. Now, calling them pronucleus in that situation just means, okay, now we're having a precursor to the nucleus of the new potential baby, right? The new zygote. And these two guys are moving closer together. The chromosomes in there are actually starting to condense into visible chromosomes, and we're starting to set up a mitotic spindle already. And then fertilization is really when these two pronuclei mix and all their chromosomes come together to initiate the first regular cell division of the fertilized egg. So this is where fertilization happens, when you start mixing the chromosome material from the sperm and the egg, not when the sperm gets in. So that's important to keep separate. Sperm penetration is what triggers the egg to finish meiosis. Fertilization is not technically until all those chromosomes mix up in the middle. Okay, so that's what fertilization is. And so here at step 4, chromosomes of pronuclei mix, fertilization is accomplished. And then we almost immediately start setting up for the first cell division of the zygote. And zygote is the term of this fertilized egg. Okay, so a zygote, and that's not just a term for humans, that's for all animals. Fertilized eggs are zygotes. Okay, so we're clear about the difference, sperm penetration versus fertilization. And then all that sperm penetration stuff, make sure you know about the acrosomal reaction and the cortical reaction, what goes on there, why it's important. Yes?
So, I'm just curious to know, does every single sperm have different character traits that you test on? Or do you have a limit of what?
You have a limit, but that limit with 23 pairs of chromosomes is very high.
Yeah.
I forget the number now exactly, but we'll do that in chapter 29. Do the math. I could look it up. I think I have all my notes here. It's really high. Where's my? This is the probabilities. Let me see.
And does the egg also have that number?
Yeah. It's just that women don't release that many eggs. Okay. You can make 8.4 million different kinds of sperm. And you can make 8.4 million different kinds of eggs. Obviously, you're not making that many in women anyway. So the chances of, if you have the same pair, the same man and woman have multiple kids, there's no chance, really statistically insignificant chance that these kids are identical genetically. You know, because you have, what was it then, 8.4 times 8.4 million leaves you with 71 trillion types of offspring with one pair mating.
Wow. So when you tell someone they're 1 in a million, they're really 1 in a 7 point.
Oh, you are 1 in 8.4 million. And then if you look at your mom, you know, that's another 8 so you're really 1 in 71 trillion.
Yeah.
Just with your set of parents.
So kind of special. Thanks for that.
I know, you're special. Yeah. We're all. You know, that's why you can have kids. You know, some people have 5 kids and they're all really different in their behavior, their looks, and all that because of this big variability. But more on that later.
Yes.
Right? Okay. So we'll look at. Hopefully we'll get to that. I'm trying to get to that. That's why I cut out development, because I think that's kind of fun too to have a little bit of that chapter. Okay. So fertilization. We're good in fertilization. This is a picture of actual pronuclei at fertilization. So there are the pronuclei. Here are some polar bodies. That's the pellucida, the eggshell. You see how it made a gap? Okay, in vitro fertilization, they actually inject sperm into the eggs because that hole doesn't really work as well in the lab. So a lot of times they'll help the sperm to get in there to fertilize. Okay, so now what happens? The process. So fertilization happens way up there, right? In the uterine tube, in the fallopian tube. We are then traveling for about 7-8 days down the fallopian tubes until we reach the uterus. And during that first week post-fertilization, there's already some embryonic development, or in your book they call it pre-embryonic because you don't have precursor organs yet. But I don't see us needing to make that distinction. So this is just an early embryo. In animal biology, a 4-cell embryo, you would call that a 4-cell embryo. So I don't see the reason to call it a pre-embryo versus embryo. Just call it embryo. Now, the process that happens first, once that the zygote has formed the fertilized egg, is called cleavage. And cleavage is the process of mitotic cell division of these early embryos. And in humans, the cell divisions are pretty equal. So you take the zygote that divides into 2 cells, then those divide, and you get 4, and then 8, and then 16, and 32, and so on, and so on. At some slightly later stage, it gets a little asynchronous, so they don't all stay on that same track, that they all divide exactly at the same time, but all of that is cleavage, right? The cell is cleaving into daughter cells by mitosis, right? Regular cell division. Now you're just growing this thing and growing the body and laying down the basic body parts. Now, it is really slow in humans. So when you look at this, 2 days you only have 4 cells. And then 3 days you got a solid ball of about 100 cells. That solid ball is called a morula. The cells are called blastomeres, the individuals, and they're still in this eggshell. They don't really show it that well, but this white thing is still the zona pellucida. Well, there's the label, okay. So it's still inside that eggshell. So you have all this cleavage happening, the cells dividing, but there's really no growth. Okay, you're confined in that eggshell, in that zona pellucida, and there's no growth. So every cell division results into smaller and smaller and smaller and smaller cells. And the cells are called blastomeres. So here, this is the same size as the initial zygote. Probably because of diffusion limiting the access to nutrients of the cells in the center of this ball, it starts hollowing out. And this is actually now a cross-section. A little bit later stage, so now we're kind of at 4 stage. The morula hollows out, fills with fluid, and then it hatches from the eggshell, and you can actually see it. It will burst open. People look at this in mice and some other mammals that you can study more easily, and it hatches out, and then it will actually fairly quickly get a little bigger, but that's mostly from fluid entering this cavity and just expanding that cavity. So the cells, there's still not really growth of the individual cells. This stage of the embryo is called the blastocyst. A blastocyst. And the blastocyst stage, this is what implants in the uterus. So usually this sort of bounces around in there for a few days, nourished by endometrial gland secretions, and then it will eventually adhere if it's in good condition. There are actually lots of them that don't make it. Some estimates are as high as 60% of fertilized eggs don't actually make it to implantation or any sort of early pregnancy. And you never know the difference because you just have a period and you don't know if there was a fertilization. So here's the blastocyst hatched from the egg, and we need to distinguish these terms on here. The outer shell is called the trophoblast. I shouldn't call it shell. The outer layer is the trophoblast. And this trophoblast is going to form the fetal portion of the placenta. The trophoblast forms fetal part of the placenta. And only this inner cell mass, this is what forms the body of the embryo and the fetus. Okay, so the trophoblast, the outer layer, and again, this is in 3D. If you looked at this from the outside, it would just look like a ball. That forms the fetal part of the placenta. The other part is from mom's tissue. So in humans, we have a half and half placenta, half from mom, half from baby. So baby's half comes from the trophoblast, and the inner cell mass, that actually builds the body. Okay? So that's where the body forms. And in people who have genetic problems or need in vitro fertilization, they often will take a blastocyst stage embryo and very carefully remove one of these cells from the inner cell mass, do genetic testing on those to make sure if you're putting early embryos in mom for implantation that they are healthy. You know, sometimes people they know familial history, they have bad genetic conditions, and they have to go through in vitro to just screen these embryos to make sure they're not getting a doomed pregnancy. So, you know, you still want to have a child and not keep it up to chance. You may want to consider something like that. And there are some conditions where that's definitely worth it. Okay. Very interesting. Okay, so, and take-home message, that's okay. So these little cells in there, they're not, you know, now predetermined, okay, if you take this one out, it won't have a foot or anything like that. You see what I mean? So they're all still able to completely, you could take more than one out, in fact. Sometimes they do that. So they do the genetic testing. There are some conditions that, you know, if it's a boy, he's going to have this. So they do sex selection and make sure they only implant girl embryos. So these things are important to be able to do for some genetic issues that people have, or they need to just say, okay, we don't have kids. Okay. So for some of these conditions, you need to do sex selection even. Not because you want just girls, but because of if it is a boy, he's going to have this bad illness. So you can do that by doing in vitro to really be able to access the embryo, take that cell out from that inner cell mass, test it, and then if that's okay, you can take that embryo, put it in mom, and hope it implants. And usually they implant 3 or 4 at most IV in vitro clinics, because any more than that, there's just a very high, you know, Octomom.
Yeah.
Do you remember Octomom?
Yeah.
Some of you anyway. Most clinics will refuse to do that and implant that many because they are just the human body's not meant to carry 8 babies.
No.
Or you can implant, you know, 3 or 4, and some people do a reduction. So you're hoping that at least one of them takes, but what if 4 take, or 5 or 6? Then you do a selective reduction and keep just 3, let's say. That's more healthy. So you can have a much healthier pregnancy. A good chance that the kids will actually make it and not have low oxygen and all kinds of other potential pregnancy issues with them, and have much better outcome. I know this is kind of weird. What time is it? Is it time to go? Is that what they're telling us?
Get out of here.
So these cells are kind of interchangeable. So you can take stuff out and they will replace themselves. And let's just do this implantation part and then we'll go. So after about a week, and remember what we saw in the uterine lining, all that stuff with the hormones we did last time, that's when the uterus is ready and you have built up the endometrium, right, in the luteal phase. A week after ovulation, we're ready to implant. Okay, let's quit. I'm sorry. A week after we're ready to implant, and we'll implant on Monday. I don't know.